Hello and welcome to today's webinar. On behalf of Drug Target Review, BMG LabTech, LabSite and the University of Wisconsin, I'd like to thank you all for attending. I'm your moderator, Ikra Farouk. Today's speakers will be Dr. Iain Ananiev, Core Facility Manager at the University of Wisconsin-Madison Carbone Cancer Center, Dr. Carl Peters, Senior Application Scientist at BMG LabTech, and Dr. Iain Russell, Senior Project Manager at LabSite. Following their presentations, we'll move on to a live question and answer session where you can pose questions to today's speakers. Please remember you can submit questions at any point during the webinar using the Ask a Question panel situated below the player. So without hesitation, may I pass over today's keynote speakers, Iain Ananiev, Carl Peters, and Iain Russell.
Thanks, Ikra. Today, I'm happy to have a chance to talk to you about how BMG is providing microliters suitable for the HTS market. The Ferristar FSX is the fourth generation in the Ferristar line. With each successive generation, we've continued to make improvements to ensure that we have a reader with outstanding sensitivity in all detection modes and can attain this high sensitivity even with extremely fast read times. The FSX can perform all of the most common detection methods. This includes absorbance, for which we have a spectrometer that can detect a spectrum from 220 to 1,000 nanometers in less than a second per well, and photomultiplier tubes, or PMT-based detection for all other modes, including dedicated PMTs for time-resolved fluorescent measurements. Today, we will be focusing on the detection of luminescence and alpha screen, which Iain will be talking about later. A key to the multifunctional detection in the Ferristar is our optic modules, which contain all the components needed for any particular assay and can include 2 emission filters for simultaneous dual emission detection using matched PMTs. Each module is barcoded so that it is easily recognizable by the reader when a module is changed. Furthermore, any module can be used in either top or bottom reading mode. Another way that the FSX improves sensitivity is through the use of appropriate excitation sources. For example, with a dedicated laser for alpha screen. This excitation source can also be used in multiplexing, such as the AlphaLISA AlphaFlex 545 example shown here. Assay performance can also be improved with BMG's unique decay curve monitoring function. This can be linked with a data analysis tool, the Integration Time Wizard, to optimize timings used for your experiment and to maximize important assay quality parameters. The high sensitivity attainable in the FSX means that low volumes of sample can be used and even the highest density well format can be employed. This is great for HTS campaigns where saving samples and saving on reagents is a key factor. As I mentioned, the focus of today's talk by Iain will be on HTS projects that use luminescence and alpha screen. A potential concern in these types of assays is crosstalk, when a neighboring well influences the signal that is detected. The effect is especially relevant as higher density plate formats are used. Therefore, I would like to spend a little time to show you how the FSX addresses these problems. This image is simply to show why crosstalk can be a concern. A standard white plate is not impervious to light. This example shows the light coming through the bottom of the plate, but you can imagine that a similar effect is seen through the walls between the wells. The first remedy that the FSX employs is a physical blockade using what we call aperture spoons. Figure 1 shows that in the absence of one of these spoons, light can come out of the top of the neighboring well and could reach the detector. In Figure 2, the aperture is in place and light from the top of the well is blocked. As you can see, there's still the possibility of light moving through the wall of the wells. For that, we can employ a correction based on an initial calibration step for your plate. Another physical change that we can make is shown here. Afterglow is an effect in alpha screen on neighboring wells by the excitation of a particular well. this effect decreases with time. So simply changing the reading pattern from something that is typical, shown on the left, to the interlaced reading mode shown on the right will reduce the afterglow effect that row B would have experienced from row A. Shown here is a luminescence crosstalk evaluation experiment using a 1536 well plate on which high-count positive controls were dispersed across the plate. As you can see in the top panel, when no crosstalk control is used, you can have very high signal in neighboring wells. But adding crosstalk control to the same plate results in a substantial reduction of this noise seen in the lower panel. An independent HTS lab has performed a comparison of the FSX to other readers commonly used in high throughput to assess performance in crosstalk determination experiment. As you can see, the crosstalk correction procedure we have implemented helps the FSX to outperform these readers based on the amount of crosstalk observed and the statistical variability of the replicate. All the technology that I've told you about today is thanks to our team of scientists. They are highly experienced microplate professionals that have listened to the needs expressed by our customers and have really delivered some outstanding solutions. The design elements that are translated by our assembly team and testing team work to ensure that every reader leaves with the best components possible. With daughter companies around the globe, we work hard to make sure that you will have the support you need no matter where you are doing your research. Thank you for taking your time to join us, and now on to Iain from LabSite.
All right. Thank you very much, Carl. My name is Iain Russell. I'm a product manager at LabSite, and what I want to present to you today is some of the recent advances that we've made with the Echo liquid handlers. Before I get into the technology, for those of you new to LabSite, I just want to give you a brief introduction to the company. LabSite was founded in 2000 and launched the first acoustic liquid handler in 2004. We followed that up in 2006 with a Echo 555, which met the needs of our high-throughput sample management and high-throughput screening customers. In 2011, we launched dynamic fluid analysis, which I'll go into in some detail in one of the later slides. And where I'm going to focus today is on some of the products that we've launched this year with the new Echo 650 series and the automation that surrounds that. So before we go into the instrumentation, let me give you a very brief introduction to Echo liquid handling technology and the acoustic droplet ejection approach that we use. What you're looking at here is a cartoon on the right-hand side of the slide in which we have a source microplate in the middle there. Below that is an acoustic transducer that is coupled to the bottom of the source plate with a coupling fluid, which in our case is water. What we can do is we can focus acoustic energy that is generated by the transducer at the meniscus of the liquid within a particular source well. And through the focusing of that energy on the meniscus surface, we can eject droplets of a very predetermined volume, either 2.5 nanoliters or 25 nanoliters, and eject those from the source well into a destination well that sits within an inverted microplate above. This is very rapid, so we typically are ejecting anywhere from 200 to 500 droplets per second, and very flexible in so much that we can position the transducer under any of the source wells and eject into any of the destination wells. So we have any well to any well capabilities. One unique feature of the approach that we use here is to employ a technology we call dynamic fluid analysis. Essentially what you're looking at here in the video is a step that takes place prior to ejection in which we do a very soft ping of the fluid that's in the well, and we look at the reflections from 3 different surfaces. The first surface is the bottom of the plate, the second surface is the bottom of the well, and the third surface is the meniscus of the liquid itself. And from that we can infer various properties of the fluid that is within the well. And the 3 major components that we're interested in are the well height, so where that meniscus lies, and that helps us with the focusing. And then also information about the surface tension of that fluid and the viscosity of the fluid, which enables us to do real-time adjustments to our calibrations. Now, I should mention that all of our instruments are shipped pre-calibrated from the factory. So what that means as a user is that it's pretty much plug and play. You will be required to select a particular fluid class, but then the instrument will make the necessary adjustments within that fluid class to accommodate changes in the fluid that may be contained within that well. So this is really quite unique, and it eliminates any operator interventions that are required during the transfer process. So essentially eliminating the need to do a calibration for the particular fluid that you're interested in. Now, we've taken this technology and we launched in February of this year a brand new solution around the Echo 655T liquid handler. What's unique about this solution is that it accommodates a new consumable type, which we call the FluidX Acoustics sample tube or fast tube. And that enables our customers now to take sample storage or compounds that are in sample storage and take those all the way from the store to their assay using a completely acoustic workflow. We, of course, still support the standard lab site consumables. So there's a variety of different microplates there in 384 and 1536 formats. We have software that supports the various applications. And then I'm not going to go into great detail, but there is also a brand new automation system that's highly scalable, totally modular, that allows our customers to build the types of solutions that they need for their workflows. In order to get to the solution, we worked with a variety of outside partners. Most notably, we worked with Titian around the Mosaic software to provide a LIMS system that is tightly integrated with the overall workflow supported by the Echo 655T and Access Automation System. And we worked with Brooks to provide the acoustic sample tube as well as storage solutions for the tube itself. Since that time, we've been working hard to take that same technology and now expand it out into a variety of new instruments, making a series of, or a family of instruments that we call the Echo 650 series. The important aspect to this is that they share all the capabilities of the 655T in so much that they can accommodate the acoustic sample tube either directly if they're a T-enabled system, or they have the potential to be upgraded to accommodate the tube at a later time in conjunction with any growing needs within your lab. They all share the high transfer accuracy and precision across the entire family. So essentially at the bottom here we have the Echo 650. That's our medium throughput system. As I mentioned, it is upgradable to support the sample tube, as well as it can be upgraded to get to higher throughput as the need arises. The Echo 655 is our higher throughput system. It also is upgradable to support the tubes. And then the Echo 650T is a medium throughput system that supports sample tubes, and of course the 655T is the highest throughput and supports the sample tube. Now, the reason that our customers purchase Echo instruments really falls into four major buckets here. The first one is to maximize their lab budget. The ability to miniaturize assays is incredibly important to getting there, as well as the fact that the Echo functions without the need for any tips, and so you eliminate the tip costs associated with that. The ability to increase productivity or lab efficiency, both in terms of eliminating any of the confounding issues with tips, so no sample loss on the tips. It eliminates any cross-contamination that's going on there. And then the ability to rapidly iterate on your assay design. So particularly if you're in a development mode, this becomes incredibly important in terms of the level of flexibility that the ecosystem provides with that any well to any well transfer. And then lastly, in terms of the automation side, this enables labs to increase overall efficiency, free up operator time to do more high-value types of work within the lab. Here's some examples of how the Echo might maximize your research. So maximizing those reagent savings. We've seen anywhere from 4-fold to 100-fold reduction in the amount of reagents that customers are using, which of course equates to a direct savings from a budgetary standpoint. And then in terms of the sample usage, many times that sample is incredibly valuable, and any way to stretch that further is important to our customers. Maximizing efficiency, on the lower left-hand panel here, you can see this is a particular example of a pooling workflow in which the customers have taken it from a 18-hour workflow down to a 3-hour workflow with a 15 hours of savings there. So quite meaningful from that perspective. And then lastly, maximizing results within the compound screening world. This is some data that came out of Bristol Myers Squibb, which indicates that once you get away from using a traditional tip-based system, then the potency of the compounds that one is screening significantly increases, and in this particular example here, the customer is able to see a 30% more hits in the library that they were screening there. So this is my last slide. I just want to pull it together here. So what I've presented to you is a family of instruments, all of which are ready to go out of the box. No tips are required with the transfer, so that eliminates a lot of the sample carryover or contamination issues that one might have there. It does enable any well to any well transfer, which opens up rapid cherry picking types of activities and assay miniaturization. In terms of the different instruments, I focus most of our attention on the Echo 650 series, which is our next generation system, which is upgradable in terms of throughput and the use of acoustic sample tubes. But we still do have the Echo 550 series, two instruments there, the 550 and the 555, which are our first generation instruments and are well proven within the various markets that we sell to. And then lastly, an instrument that I didn't touch upon at all in my earlier slides, which is the Echo 525, which is specifically designed for higher volume transfers and aligns well with our genomics and biochemical markets there. So with that, I will stop and turn it over. Thank you.
Hello, everybody. So my name is Ananiev, and I'm from the University of Wisconsin Madison, where I manage the daily operations of the small molecule screening facility. So before we begin, I'd like to introduce our facility, kind of tell you a little bit who we are, and thank everybody for organizing this and letting us present. So the facility was started in 2003, and our faculty director is Michael Hoffman. We are located in Madison, Wisconsin, which is in the Midwest of the U.S., and we operate via a chargeback mechanism. So essentially, what we do is investigators and companies come to us with projects, and we're able to do this on a contract basis where we don't hold any IP, and we're able to do the services that you want us to provide. So the first question people usually ask about our facility is, what does our name mean? So we'll spend a couple minutes just going through the background. So small molecules refers to a small molecule which is usually under 900 Dalton, which may perturb a biological process. And the important point here is that most drugs are small molecules. So you can think about it this way: if you have a protein and that you treat with a small molecule that acts as an exogenous ligand, that will affect the function. So that's, in a nutshell, what small molecules are. And then we'll go a little bit through the origins of high-throughput screening and how that relates to small molecules. So HTS really began in the mid-1980s, and the origins were in natural product drug discovery. So you would derive extracts from natural products, tested in a disk diffusion assay, like this, which is a really old school method of doing this. And you'd be doing one extract per one test. You'd need milligram amounts of compounds, and the labs that were doing this were testing about 20 to 50 compounds per lab per week, so very slow throughput. So by 90s, you have a lot of automation and multi-well plates coming online. The industry standardized on DMSO stocks as a solvents, and labs were doing about 10,000 compounds per lab per week. It's also during this time where one of the first big successes for the methodology, the creation of imatinib, comes in. And then 2000 to today, you're seeing further miniaturization with assays going into 384, 1536, and now 3456 well plates. And there's been a significant effort now to actually go back and screen natural products again, primarily as a result of the antibiotic drug resistance. So at the heart of any HTS effort is the small molecule collection. So I'll talk a little bit about our collection here at the small molecule screening facility at the University of Wisconsin Madison. So we have close to 500,000 compounds, majority of them being diversity libraries. And by diversity libraries, what we mean is these are compounds that were synthesized to be drug-like, but they're not drugs. We have about 5,000 drug-like molecules, which have gone through some degree of drug optimization. So these are all known drugs that have been approved by the FDA, and a lot of the compounds that have gone through clinical trials. And we have a small collection of targeted libraries that are specific for various purposes, such as kinase inhibitors, anti-infective, natural products, and some mechanistic diversity. So my work with the facility really began with this screen that we did with Professor James L. Keck at the Department of Biomolecular Chemistry here at the university. And this was a fluorescence polarization screen for a protein-protein interaction. Now this was a project that was set up using non-acoustic liquid handling. So this was using a Beckman FX. And just to kind of give you a summary, we screened 75,000 compounds. We used about 2.4 liters of protein solution to test for FP. And this was a fairly expensive project despite using a simple readout assay, primarily because of the tips. So what we have here is, this was actually our setup for our daily work through. So this is 25,000 compounds. You can see the source plates that have the compound, target plates, and in the back here is all the tips. So really this is a fairly expensive way to do this. You waste a lot of protein due to dead volumes, and what is especially true these days is the environmental impact of putting all these plastics in the landfill is rather bad. So working with this lab, we set out to really miniaturize these protein-protein interactions for their targets of interest, and try and do this so that we can cheaply and reliably screen hundreds of thousands of compounds in this. So this was a publication that resulted as a part of this project. We did a high-throughput screening strategy to identify inhibitors of SSB protein-protein interactions, and this was an antimicrobial target. For this methodology, the lab decided to go away from FP and do screening for protein-protein interactions using alpha screen or alpha ELISA. So this is a Perkin-Elmer assay, which replaces an ELISA. It's a bead-based proximity assay. So you have your analyte of choice and two beads conjugated with antibodies against it. And if there is an interaction, once you excite one bead, bead, it releases oxygen and creates light. So this is a very fast assay. It's only a 1-hour reaction. And as we found out, it's scalable to low volumes. So currently we're running this at 3 microliters in 1536 well plates. So one of the main aspects of cost reduction for HTS is reducing volume and tips. So for this, 1536 well plates provide a fairly, I don't want to say ideal, but a fairly good solution. So as you know, volume used scales was the surface area. So here we have 48 well plates, 96, 384, and here the little red square is 1536. So this just puts you in perspective the surface areas we're working with. And the majority of the cost savings here comes from reagents. So this is about an order of magnitude reduction in use based on compared to 384. So for a 96 well plate, you'd be using about 50 to 100 microliters of reagent for detection. In 384 well plates, using 20 to 60. For 1536, it's really 3 to 5. So then the previous two sections of this dealt with some of the instrumentations that can be used for this. So the way that we went around setting this up is you can see the general scheme of the assay here. So this shows the specific proteins, the interactions with the beads, and the assay workflow. So the workflow for the high throughput screen was we have an Echo 550 instrument, which we used for compound addition, which was a 10 nanoliter volume. We use a Mantis, which is a small liquid, tipless liquid handler for adding reaction master mix, which is 3 microliters, and then a BMG Ferrostar for detecting alpha lysa in 1536 well plates. We can go a little bit in the nuts and bolts of how this assay runs. So the really critical part of this is setting up the timing of the plate reader to work with the timing of the liquid handling addition. So currently to fill a 1536 well plate with the Mantis, it takes us about 20 minutes. And with the settings that we use to detect the alpha lysa signal, it takes us about 30 minutes to read the plate. So the Ferrostar has a couple of fairly interesting features, such as dictating the direction of how you read and the settling time. So the direction of how you read is set up so that it mimics the direction of how we're adding the liquid. So the liquid handler here adds starting at the bottom right-hand corner going up. So we have the plate reader mimicking that to minimize the lag time. And also the settling time is really dictated by the signal strength. So for alpha screens that have produce a lot of signal, you would set the settling time a little bit higher. For this current one, we had it at half a second. So this is just a snapshot of the data as it comes off the plate reader for this particular project. So obviously you have a positive and negative control. In this particular case, we're screening for inhibitors, so the positive control is a low signal, negative control is just DMSO. We do run an IC50 curve of our positive control on every plate. And what you might notice here, if you're really careful, is there's a bit of an edge effect, which is a known issue with 1536 well plates. So if you are interested in how we dealt with it, I would recommend you read the SLAS discovery paper. But without going into the specific detail and nuts and bolts about it, what we did is we actually solved this problem computationally. We tested plates full of DMSO with just the assay, and then based on that, we have a scaling formula that corrects the edge effect on every well. So if you remember the initial screening setup that I showed you for 384 well plates, this is the summary of our alpha lysa screen in 1536 well plates. So we screened a similar number of compounds to FP, 72,000 compounds, but we only used 240 mils of protein solution, so that's an order of magnitude reductions. This is a Z-prime figure, so all of the Z-primes for the plates are above 0.5. And what's most importantly for an academic lab on a cost-saving perspective is, so the standard alpha lysa reaction is about 25 microliters, which gives you 10,000 data points per 5 milligrams of beads. We were able to reduce that 41-fold and screened about 300,000 data points for an equivalent amount of reagents. Also, we similarly, which is a huge savings for the grad students and everybody involved with this, is we were able to minimize protein use to less than 5 mg for the entire screen. And the estimate for running this as FP would be about 1 gram. So these savings are very significant. All of this data is available on PubChem if you want to look it up. And since publication, we've screened over 500,000 data points using this methodology for both for James Keck's labs and for other labs in different projects. So this has been a fairly routine assay for us now that the methodology is established. So now I'd like to kind of switch gears and talk a little bit more about reporter gene assays, so cell-based screening in 1536-well plates. The way that we have done several of these efforts is using reporter genes. So traditionally you'd use a GFP or a luciferase. Once you start switching into 1536-well plates, the brightness of your reporter becomes critical. So GFP is unusable, and then Promega, which is a biotech company here in Madison, has several luciferases on the market which are used for high-throughput screening efforts. The one that we used for this screen and the one that we recommend to use is Nano luciferase. It's very small and very bright, so it works extremely well in 1536-well plates. So the project that we ran for this was for the laboratory of Xinyu Zhao at the Weizmann Center at the University of Wisconsin Madison, and this was for establishment of reporter lines for detecting Fragile X, FMR1 gene reactivation in human neuronal cells. So in this particular screen, FMR1 is a gene that gets silenced in Fragile X syndrome, and their lab was interested in finding activators of this gene, so basically something that would be able to correct the gene expression defect by reactivating the gene. To this extent, they constructed a cell line with a nanoluciferase reporter knocked into it using CRISPR-Cas9. What was interesting about this construct is that it still produced both the FMRP gene and the nanoluciferase. They constructed this. There was extensive characterization in both mRNA and protein expression levels of both FMR and nanoluciferase. And so what's interesting is, so the screen was done using human neuronal cells, which were derived from iPS cells. So this is actually a fairly intricate differentiation protocols where you would take human iPS or human ES cells, and through various media changes over the span of about 2 weeks, differentiate them into neuronal precursor cells. And here what you can see is that with the two cell lines that were established with the nanoluciferase and also with human ES cell H1, you can see that Nestin PAX6 are expressed. These are markers for neuronal progenitor cells for this particular project. So the interesting thing is there is a compound, which is a DNA methyltransferase inhibitor, 5-azacitidine, which can can reactivate the FMR gene in iPS cell-derived neuronal progenitor cells. And this is the profiling of the cell lines that they derived. So this is FMR1 nanoluciferase expressing cells treated with 5-azacitidine, and you can see that both FMR and nanoluciferase are upregulated as a result of this treatment. So at this point, they came to us trying to set up a high-throughput screening campaign to find more compounds that are able to reactivate gene expression of this gene. So the first step that we did was to take the cell line and set up an HTS screen in 384 well plates. So we worked out the various conditions where you would plate both cells and matrigel at the same time into 384 well plates, add the drug library, then add NanoGlow and read. Initially, we tested a small library of 128 known epigenetic of compounds that are known to modulate epigenetic expression. So, what we did find, which was a good confirmation for us, is that both 5-azacitidine and a 5-aza-D-cytidine worked. So this was very encouraging. However, once the lab that we were working with, so Xinyu's lab, started looking at the amount of cells that they would need quickly became obvious that a large HDS campaign would not be possible in 384 well plates because of the difficulty of differentiating the cells, the limited amount of biological material we have. So they really pushed for it, and we worked with them to optimize and put this screen into 1536 well plates with the primary motivation of saving cells. So this is a figure from the paper showing some of the initial work for optimizing the 1536 well HDS. So this is just a control plate with 5-azacitidine and DMSO Z primes that we're able to get. The cells survive and look really nice. And this here is a screen at 4 concentrations of all known FDA-approved drugs. Again, the only thing we picked out of it is 5-azacitidine. So since the publication of this paper and since this methodology was worked out, we've screened about 330,000 compounds actually on this project. So the plate reader setup for this was actually fairly simple, and since we're looking for reactivation, the majority of our plates have very low signal, so none of this crosstalk or anything came up as an issue. So again, you just have negative control, positive control, data gets collected, we look for activators. The timing is pretty straightforward here because we're using a multi-drop to add all the reagents, so you don't have to worry about it quite as much. So to add the concluding remarks about the 1536 well screens that we do at the SMSF, so we've now done efforts for protein-protein interactions that we've shown you an example in. We've done cell-based reporter assays. We've done biochemical screens for various enzymes using both fluorescent and luminescent reagents. And we've actually now dwelled in doing some cell-based dual reporter for both nanoglow and renilla and firefly luciferase in 2 wells. So the instrumentation that we have for doing 1536 well is Echo. We've now added a blue cat biowasher primarily for trying to multiplex more to be able to remove liquid from 1536 well plates. We have the BMG Ferrostar and Amantis. And really the big motivation for us is throughput and cost savings and saving biological reagents. And again, to end this presentation, our facility is part of the bigger drug development core on the University of Wisconsin-Madison campus here that has 3 components. So we have the small molecule screening facility, we have a cancer pharmacology lab, and we have a medicinal chemistry lab. So we're really able to do most of the initial preclinical drug development efforts here in-house. And if you have any further questions or you want to discuss any potential projects with us or any questions publication, this is my information and this is our website. All right, thank you.
Thank you, Jean, Carl, and Ian for your excellent presentations. Before we move on to our live question and answer session, I would like to invite you to provide us with some feedback on today's webinar. In a moment, a poll will appear on your screen asking you to rate the webinar. If you could take a moment to provide your feedback, it would be much appreciated. We now have the pleasure to introduce our question and answer session, where today's speakers will answer all of your questions. Don't forget you can still submit your questions using the Ask a Question panel situated below the player. Let's take a look at the first question. Will you buy Shellac Chemicals bioactive library?
Sure, we have several libraries from Shellac. We like them, they're a good company.
Okay, thank you for your answer. We have a similar assay using cells stably transfected with nano-Luc tagged protein. However, we have very poor Z factor, less than 0.3 on a 384 well plate. We have tried many reader with similar results. We have tried to increase the amount of cells per well without any luck. Do you have any thoughts on this?
Yeah, I mean, the major thing I would really try to optimize is the amount of time that you let the luciferase get expressed and how long you grow the cells for. Also, increasing the number is probably not necessary. You might want to actually try and bump that down a little bit so that the cells are happy and dividing.
Okay, thank you for your answer. The next question. Yeah, no, go on, sorry.
If I could chime in on that a little bit. As far as from the reader perspective, I mean, I would of course need to see some data to, if you want to have this discussion more offline. But there are sort of three things that go into the Z prime, and that is the signal to noise, and then variability. So it'd be interesting to see which of those factors seems to be playing a role, and then we could make some recommendations based on that, how to possibly, from an instrument setting point of view, to improve it.
Yeah, and I want to add on that. I think that's a great point, because if you're seeing a lot of variability and scatter, you might have clumped cells, and it might be a more of a tissue culture problem. Whereas if you don't have enough signal to background, it might be the protein expression issue. So.
Thank you for your answers. The next question asks, what is involved in the crosstalk determination?
Yeah, I think that's a BMG question. We just use the instrument settings on our end.
So, I think the presentation went over this to some extent, but as far as the instrument-assisted corrections that were referred to, there's a wizard that takes you through step-by-step where to plate high and low signals on the plate, and then to basically, as I indicated, to perform a calibration that would allow for the variability to be decreased. So it really just comes down to plating high signal next to low signal and making that comparison.
Thank you for your answer. Are there any other remedies that can be attempted to improve crosstalk in luminescence and alpha screen assays?
Again, I think that's more of a plate reader question, but I can answer a little bit on that. All the previous suggestions are still valid. One of the things we had luck playing with is the settling time parameter on the plate reader. So if you just simply wait before you read the next well, that sometimes can give you a little bit of help on that.
Yes, the only thing that I would add is, this is Carl from BMG, that the plate type does seem to have an impact. So there are variety of different vendors that have that have different types of formulations for their plates, and that seems to play a role. So possibly evaluating the plate type that you're using is another thing that can improve your results.
Thank you for your answers. What range of volumes work for the 1536 well alpha screen experiments? What is the minimum and maximum volume that can be read in those plates? And what is the smallest volume of alpha screen reaction that can be read in a 384 well plate?
Yeah, so that's a great question. So in 1536 well plates, it's pretty much 2 microliters to 10, but 3 works a lot better than 2. With 2, you can run into some issues with meniscus. So it's really between 3 and 10 microliters for 1536. And for 384, depending on what plates you use, if you use I think they're called the Opti plate from PerkinElmer, so these are 384 well plates, but the well size is the same as 1536, so there again it's 3 to 10. Otherwise, you would probably need at least 20 microliters in a standard 384 well plate.
Thank you. The next question is on the order of addition. How stable are library compounds when you add them to dry plates? Is there evidence that plastic absorbs the compounds?
Compounds are extremely stable, and I don't think there's any evidence that it absorbs to plastic. I mean, we've shipped these guys globally with wonderful results.
Thank you. The next question asks, can we conduct fluorescence polarization assays with intact bacterial cells?
Yes, I kind of wanted a little bit more info on this one. I think in theory, it's probably possible. From a plate reader perspective, that'd be a good question for BMG. We certainly have not tried doing that in our lab.
This is Carl from BMG. I don't know of any examples of this being done. Any type of cellular fluorescence polarization is difficult mostly because of the background fluorescence that is present within the cells. So I think that the first consideration would be selecting fluorophores and tags that are going to be more in the red range rather than the green range that where you can have some interference. So then beyond that, then it would be whether or not you are reading the right part of the well for the bacteria that are actually moving around within the well. It could be a confounding factor.
Thanks for your answer. The next question asks, when reading luminescence in 1536 well plates, is there any bleed through between the walls? If there is bleed through, how do you address the issue?
Yes, I think this has already been answered in a couple of previous questions. So yes, there's bleed through, and we, especially for the alpha screen one, we addressed it computationally. We ran some empty DMSO plates, and there's some crosstalk correction built into the plate readers. I don't know if Carl wanted to add anything.
Thanks for your answer. The next question asks, Echo is good for nanoliter, but what do you use to transfer into microliter, around 10 to 20 microliters?
Yeah, that's a great question. So we use a Thermo Fisher combi drop. There's Biotech makes. So there's various barostatic pump-based instruments. We also use the Mantis primarily for things in hundreds of nanoliter range and low microliter.
Thank you for your answer. Unfortunately, that's all we have time for today. Any unanswered questions will be responded to via email. Please join me in thanking Jean Ananiev, Carl Peters, and Iain Russell for their time today. On behalf of Drug Target Review, BMG LabTech, LabSite, and the University of Wisconsin, I'd like to thank you all for attending today's webinar. We hope to see you again soon. Goodbye.